I'm going to discuss multiomic identification of novel smooth muscle types and human coarctation of the aorta. It's a very exciting subject, just in case you were wondering. And I'm also going to multitask a little bit. When I signed up and planned this talk about a year ago, I thought I'd just talk about my science. But obviously, over the course of the last year, the world has changed very much. And the tone of the scientific community in the country has changed. And so I'm going to multitask a bit and discuss some things that are relevant to all of you in your training and us as faculty as we go through. So how many are residents and med students? You're all mainly med students for the most part. Okay. And then I'm sure there's a different contingent out and about. Okay. To advance, I'm clicking it. Okay, there we go. So what is multiomics exactly, and what does that mean? So omics refers to the biology disciplines whose names end in omic. It's very original. So genomics is the study of genetics, proteomics, the study of proteins, transcriptomics, transcripts, or mRNA. And they are the many techniques used to characterize a process. So you integrate the different aspects of the central dogma, right? You remember the central dogma from Francis Crick in 1958. DNA becomes RNA, becomes protein, and this is how the world works for the most part. Technology has advanced significantly in the last 15 years so that we can actually quantify, characterize these different parts of the process. And that allows us to have greater insights into different areas of biology. And so that's my science portion talk of today. But what's a surgeon scientist? So a surgeon is obviously all of you in the room who are training or are already surgeons, but the combination of being a surgeon scientist is someone who is actively involved in research while having a practice, operating as well. And they bridge the gap between what happens clinically and what's happening in the lab. And I want to discuss that because it's a very important thing at this moment in time where our colleagues who are basic scientists are really under threat. I don't know if any of you have ever worked in industry where you're sitting in a room with a bunch of people who have been laid off, but I never have. And at the moment, our research meetings downtown in FCRI, Seattle Children's Research Institute, I'm sitting with a bunch of very senior faculty who have been lifelong PhDs, multiple R01s, each of them, who are worried about losing their jobs because of the way that the government is funding things. And so it's very incumbent on us as those clinicians who are also on the other side of the bench to bridge this gap even further, okay, to protect the future of American science. So these people were very influential in my upbringing, either directly or indirectly. So here is Norman Shumway, in case you don't recognize that face, father of heart transplant, father of most of the chiefs of the West Coast in cardiac surgery. Michael DeBakey, the first person to create an artificial heart. Denton Cooley, first person to implant that heart, in case you don't know that drama from before, also the founder of the Texas Heart Institute. Craig Miller, the person who really pioneered TVAR, is a huge figurehead in Stanford. My former chair and boss, Joe Wu, who is a tour de force with regard to bringing up surgeon scientists, training them, putting them out to the community. And Pedro Del Nido, my chief at Boston Children's, Del Nido cardioplegia, what most of the world uses to arrest hearts and go on bypass, was invented by him. And there are many valves in development along the way. So these are people who are actively involved in the OR, either run services, have run services for a long time, but also are at the bench developing things. So who am I? I'm Christina. I'm a congenital heart surgeon at Seattle Children's. I'm an assistant professor here at UW. I started in 2021. I have an adjunct appointment in mechanical engineering for some of my bypass work that I do as well. But those are all my little titles, but like, what is a congenital heart surgeon? I know many of you are in general surgery or maybe in some subspecialty at this point. A congenital surgeon is someone who operates on baby hearts. So this is a normal heart. You have systemic blood flow that's had all its oxygen removed, coming back to the heart, going to the right atrium, to the right ventricle, and then going to the lungs where it gets oxygenated, comes back to the left atrium, goes to the left ventricle, and then goes out the body via the aorta. But there's a whole host of children that are born where this is not the case. So hypoplastic left heart, babies that are born without a left ventricle, how do they survive? Those who are born with transposition of the great arteries, where there's actually two parallel circuits where the oxygenated blood gets sent back out to the body, gets stripped of its oxygen and just keeps going. And the blood that's oxygenated keeps circulating through the lungs, through the heart, through the lungs, through the heart. How do those babies survive? Truncus arteriosus, where you have a common trunk for the pulmonary and aortic. These are all congenital heart diseases that we fix. And when we say congenital, we really treat everyone from preterm, fetal, through like depending on your children's hospital, early 20s, 21, 23, however. And then there's a whole growing contingent of adult congenital. Dave Mockley, sitting right here, my partner, who does all the adult congenital here at UW for the most part, deals with those patients that grow up who've had their repairs as a child, who have now survived and now are adults and need a ton of very specialized care. And so that takes the rest of the population. So this is the world of congenital heart surgery, a mix of the two aspects. And you don't see us very much because we live down the street at the Children's Hospital for the most part. So unless you rotate through there where there's not too many residents that come through our service, you won't see us, hear us, or know we exist. But we do, and there's 4 of us who serve the WWAMI region. Washington, you know what WWAMI is, but there's 4 of us, so we get all the cases from all of that area. This is me. These are 2 kind of stock photos I have of myself that are important. This is me doing a cabbage, operating side saddle. I'm like 8.5 months pregnant during adult fellowship, and this is me operating with Del Nido during fellowship, congenital fellowship. Also, you can't see it, but extremely pregnant as well. And just to show you, that's my life, and that's how it goes. I come from Los Angeles, down south, so obviously I have a bit of a warm weather way to me in all my stuff. I ended up going to Boston to engineering school, where I was really taught how to think, how to formulate hypotheses, and how to get my ass kicked for the most part. And it was very formative in my preparation to be a surgeon. I worked on a vaccine at the Pasteur Institute during college, because when I was growing up in the 80s, I thought the biggest threat to humanity was AIDS growing up in LA. So I spent some time working on a vaccine. But after college, I returned home to LA County, where I did medical school and general surgery and stayed for about a decade. And then from there, went up to Stanford for cardiac training, and then on to Boston Children's for congenital training. And then here, I'm taking my first job. So if you want to feel better about yourself, my training pathway was 4 years of undergraduate, 4 years of medical school, 4 years of G-Surg, and 2 years of research in that. And I actually shortened that and hopped out to go up to Stanford and did 4 years of adult cardiac and then 2 years of genital. So I was a PGY12. And I'm triple boarded, and I'm a very proud 249th certified congenital heart surgeon in the US. There's not many of us. There are more NBA players active current than there are congenital heart surgeons here. So that's who I am. But what am I trying to do? So I'm trying to understand the why of congenital heart disease. Oftentimes you do an operation, you let your family know that everything went okay, their kid's going to survive X, Y, or Z. And the only thing they want to know is why did this happen? And I have no answer for them. Like we don't know. Unfortunately, we don't know. It hasn't been figured out. No one understands why some tissue grows and other tissue doesn't. There's no animal models for any of this. There's very little advancement in the science of congenital heart surgery, I think, because you saw that training pathway. By the time people get through, I think they just want to operate and live and do stuff. No one wants to be at the bench trying to figure this stuff out. So there are very few people around the country who are actively trying to figure out congenital heart disease at the basic level. And what do I want to do? I want to improve outcome. The operations that I'm going to talk about were, the first one was done by Crawford in 1944. It hasn't really iterated that much since then. That is massive. What technique do you know that is almost the same 75 years later. We don't really have any more insights as to why it occurs. And then once we understand what causes congenital heart disease, in particular coarctation of the aorta, you can work towards preventing these lesions rather than just operating on them. So what is coarctation of the aorta? I've mentioned that before, so just a refresher in case you don't remember from medical school. Coarctation of the aorta is when there's a narrowing at the level of the isthmus. So just after the takeoff of the left subclavian, where the ductus inserts, there is a pinch that can occur after birth. And you'll see this as the baby is born, prostaglandin from the placenta is taken away, and the ductus will constrict, and you're going to have a narrowing, and you're going to have a baby that otherwise seems to be fine, except there's a blood pressure gradient between their upper and lower extremities because they're not getting enough blood distally. And so why would I want to study coarctation of the aorta? Well, I think it's interesting. Coarctation, I think, is one of the linchpins of congenital heart surgery. It is the last left-sided lesion that's part of a host of other different syndromes. And it represents embryologically a very important zone. It is where the sixth pharyngeal arch meets the aorta. So it's a compartment boundary. And so you have a normally formed ductus that meets a normally formed aorta, but in some kids, there are some other cells in there that are causing coarctation of the aorta. Is it just that the ductal cells have invaded the aorta and are doing their normal job and are normal cells? But how did they get there? Why are they there? It's a very dynamic region. In utero, the ductus is open. That's its normal physiology. The minute you're born, that starts to close. It doesn't have its signal of prostaglandin. If you happen to have left-sided lesions or what we call ductal dependent lesions and you need your ductus to survive, you can actually administer prostaglandin and open that up to keep babies alive. So the idea of these compartments, these tissue boundaries is something that I don't think is very mainstream. It's a very vascular biology sort of concept, but essentially at the most basic level, we're all made up of tissue boundaries embryologically, interfaces where one cell's telling the other that this is my spot, that's your spot. And defects occur, so I think in congenital, where that gets mixed up. Second heart field on the ventricular septum, there's a right side and there's a left side. VSDs are one of the most common or are the most common congenital heart defect, and it occurs where those two boundaries meet. Coarctation is one of those other boundaries. So that's why I think it's interesting. And it's important. So it's one of the most common congenital heart lesions, and it's the most common disease of the aorta in about 1 in 2,500 live births. And recurrent arch obstruction is a significant contributor to morbidity in congenital heart surgery, specifically in Norwood's. This is one of the papers that we wrote once I got here, looking at the impact of coarctectomy in the Norwood population. So Norwood is, for those patients that have hypoplastic left heart, they have a 3-stage palliation. The first stage is called a Norwood. If you've ever heard that, it allows them to survive for the first couple months of life, and then they have a bidirectional Glenn. And then around 4 or 5 years, they have a Fontan operation. And this allows babies that are born with single ventricles to survive. But if you don't cut out that coarc, they have a lower reintervention-free survival, and they have a lower transplant-free survival. So they don't do as well if you don't cut out this region. And the reason why we'll see in a little bit. One of the reasons I'm studying it, because it's possible. So it's very hard to study congenital heart disease, right? You can't just take pieces of baby hearts are very small. You can't just take a little here to see what the problem is. But coarctation, we naturally cut out that area. The disease portion is actually cut out, and so you can study it. And so because there's very few animal models of congenital heart disease, no one's been really able to recreate some of these complex lesions. This is a very opportune area where we can actually study human tissue to try and figure out what the signal is. So I started work like this when I was at Stanford, when I was a fellow. I had a small lab while I was a fellow. And then when I got here, I continued it. but we started a cardiac tissue bank shortly after I got here. And so we collect the tissue that we excise from our patients in order to bank it, in order to do future studies to try to figure things out. So this is the team. There's always a team, right? It's not one person, it's a team. And so this is my team. So here's Marty Ross. He's one of my data analysts. Akshay Venkatesh is my medical student. He is a rising third year who is interested in cardiac surgery and has been in the lab for a couple of years. David Hahn is one of my collaborators from UConn, who's an expert in proteomics, who's from Seattle. His mom is here, and so as he comes out here, we do projects together. Jeff Traeger is the person who holds it all together. He's my lab manager and keeps the lab going while I'm operating all those other days a week. Amy Learston is another analyst on our team. And then this person is an important person. This is Mark Majestic. He is my collaborator. So if you want to be a surgeon scientist and you think that you're going to operate most days of the week and then be in the lab by yourself without any help, good luck. It's not how it works for the most part. So you have to find people who you're going to work with who can help and mentor you. It's the same thing as surgery, right? You don't learn surgery because you read a book and you start doing it. You don't learn how to run a lab all by yourself, right? Mark Majesty has been critical to my matriculation as a scientist. And that wasn't prescribed, that just naturally happened from having similar interests. But mentorship in that way is super important if you intend to pursue both areas and in general for life. And so I'll talk about that in a little bit, but running a lab is kind of like running a small business. It's you didn't realize, like you sign up, I have to sign off on people's like how many hours they work, you know, what the pay is, how many benefits, all these sorts of things, which you would never really think of, which I didn't really think of and didn't understand before starting one. So let's go to the science at this point. So the hypothesis is that coarctation of the aorta is the result of a breakdown of its compartment boundary. So it's between cardiac neural crest cells of the sixth pharyngeal arch. Those are the things that become the ductus. You'll see those in purple. And the paraxial mesoderm derived smooth muscle cells of the aorta. So very simply, the aorta smooth muscle cells are in red. The purple are the ductal smooth muscle cells. And our hypothesis is coarctation occurs because signals are not being transmitted and those cells are coming out and embedding into the aorta. And we see that when there's loss of PGE. So the goal is to characterize the genetic, proteomic, transcriptomic signal of coarctation of the aorta in the hopes that we can delineate the pathobiology of this region and potentially identify a cellular marker. So coarctation of the aorta happens in 1 in 2,500 live births. The WAMI region is very big. If you are born in a small village in Alaska and you have coarctation of the aorta, by the time you make it to Seattle Children's, you may be in extremis. You're born in Montana, rural Montana, that sort of thing. You can have your baby get sent home and then later find out that they have coarctation of the aorta. And until you get prostaglandin into that kid's system, they're at risk of essentially causing ischemia to their lower body, okay? They'll have neck, you know, death, everything that's possible. You happen to live in Seattle, not as big of a deal because there's children's hospitals, there's other hospitals that are right here. You have access to that. So it's very important for dealing with this around our region, around the world, that if there was a biomarker that can distinguish this before kids go home, that would be very useful for preventing morbidity. So the way we're doing that is really spatial transcriptomics. So in its most basic level, to try and explain to you, you can have tissue that can have hybridization with immunofluorescence, essentially antibodies that are labeled with a dye fluorescence that will bind to protein or mRNA, which can then be sorted and tagged so that they can be then spread out on tissue. It's the best way that I can kind of explain it. There's proprietary technology that does all of this, and then you can sequence. So essentially what it allows you to do is it allows you for the first time in the last, you know, this technology came out like 7 years ago, essentially you can see genes on tissue. You can see protein expression directly on the tissue from different probes that you're putting in. So there's a lot of pre-processing that you have to do, but this is the basic workflow. Essentially, you have a tissue sample, whatever it is. For us, it's coarctation of the aorta. You can see the histology. And then you're going to be able to detect the transcript, the mRNA on the tissue in order to see what's being expressed. And then you can look at the things at protein level, because just because you have mRNA doesn't mean that all of that goes to become protein. There's a lot of editing that happens in between those two processes. And then there's all the validation that we're going to do. So how does this work? Okay, so to do this initial work, we started with 4 samples of coarctation of the aorta. They were from neonates that were born in Seattle Children's, where we collected tissue. After birth, you can see these are their echoes, where you can see that there's obstruction. These bends, anything colorful on Doppler indicates turbulent flow. You can see this is a CTA, which shows the pinch in the aorta. And so we collected tissue from these patients, embedded it in paraffin, and sliced it. So this is coarctation of the aorta. This is an example here. And you can see this is the lumen of the aorta. This is the ductus I'm holding on that end. And you can see the narrowing through the aorta. So this is what kids are dealing with. It's very tortuous on the inside. This is this sample sliced over here. So you can see that this is the ductus. This is what I call the transition zone. Essentially, this zone that's in between the normally looking ductus and the normal aorta. And then this is the aortic lumen. And you can see here that the ductus has somewhat disordered appearance of their cells. There's the Van Giesen stain that shows collagen. And then the transition zone is even more disordered. You can look at the aorta, and it's tightly arranged, nicely oriented fibers to kind of understand the differences between those areas. But I can't see, what the difference in the protein expression are between those two things. So that's where we started to do transcriptomics. And when you look at those areas and you look at their mRNA signals, you can analyze them. So this is a lot of very, very big data. And so principal component analysis is a way to orient that data so you can understand it on a less complex level. So you're taking the most important factors and putting them on different axes in order to determine if there's differences and that you can see for the most part. So it reduces the dimensionality of data. So you can see here that the aorta is different than the transition zone and the ductus. But also on the y-axis, you can see that the transition zone is unique from the ductus. They're not the same thing. It's not just ductal cells that are in there causing this problem. And you can see that as well. This is a heat map of their gene expression. And the genetic signals between the aorta and the ductus and the transition zone are quite unique. But as well, in between the ductus and this transition zone, and let me be clear, I made up the transition zone. There's no transition zone. Like, this is a term I came up with to describe the transition from the ductus to the aorta, because it's not recognized as a unique zone. But it's genetically distinct. And so that's what we're looking at. So then if we were to continue, so when you look at ductus versus aorta, like I said, there are a lot of genes that are quite different that you can tell. And mainly it has to do with the structure of the aorta versus the ductus. So not that many of you have probably felt a neonatal ductus or the aorta, but the tissue quality is extremely different. You know how maybe in general surgery, there's that saying, eat when you can, sleep when you can, and don't mess with the pancreas. Well, the ductus is the pancreas of the congenital heart world. It will fall apart on you. It will disintegrate. It doesn't have the same integrity that the aorta has. And so clinically, I know that, but now we know we can see that on the protein level and the genetic level. And so they're quite different because it doesn't have as much elastin as the aorta. And it has some other signals that are important, like tenacency, thrombosfondin, and SALT1. This is, so when you evaluate mRNA, there are large data banks that everyone kind of publishes what they found online. And so there's a gene ontology data bank, Go data bank. And so this sorts all of those signals into the most common processes that they see. So collagen organization, blood vessel morphogenesis are very highly regulated in this. On the ductal side, protein modification and division are much higher. And so this just shows, in its most basic level, the differences in the gene expression between the aorta and the duct. Okay, so now looking at the transition zone versus the aorta, again, very similar differences between those two. This is where most of my focus is understanding the differences between the transition zone and the duct. They're not quite as different as the ductus and the aorta. You can see there's like 200 differentially expressed genes between the ductus and the aorta, and another 100 between the transition zone and the aorta. But between the transition zone and the ductus, they're much more similar. So understanding these differences are very important. And so when you do that, you can then overlay your tissue. So this is the spatial aspect of this. I know this is very broad. We'll go into them in more depth so you can see. But these are different sections of coarctation from different patients. And there's multiple ones on the slides because let's be clear, this technology is very expensive to do. So I stack as many as I can on one slide. So this is histology, right? This is like from path. You can see this tissue, it's stained with H&E. This is at fluoresced, and then our regions of interest, the areas of tissue that I'm interested in that I zoomed in and showed you earlier are there. And then here, these little color dots are different expressions of different genes. And what you can see from afar, and when we look up close in a second, that there are very characteristic signals for certain genes for certain regions, and they're consistent. So the ductus is different than the aorta. The transition zone is different than the ductus, okay? And so that's looking at very high level. And so I'm just gonna look a little bit, show you a little bit more in depth. So CCN3 is a protein that's important in cell growth, migration, and differentiation. And you can see, so this is a coarctation segment. You can see these two little punctate things here. Those are where my sutures were that I cut out. This is the ductus on this side. This is the transition zone, this area that's not aorta, it's not ductus, I don't know what it is. And then there's more normal aorta. And what you can see is that the expression of CCN3 is higher in things that we think are more normal aorta, and is lower in what we think is ductus, okay? And that's consistent on another sample here, where there is more normal aorta, okay? This was the control. And so this is more normal aorta. And then just going through this, you can see that it's consistent across all of the other additional patients as we go through. And I did this additional study because while I saw differences on 3 patients and very statistically significant and has high fold changes, I don't want to say anything from 3 patients. That's very, even though the technology is supposed to be great and everything, no one wants to go out in the world and say, oh, there's a new region of the heart that does this based off of 3 patients. So being an insecure scientist like that, I wanted to validate. So I got more patients, repeated the samples to confirm what I thought I was seeing. And so that's what's happening here. And so these are just examples of all the different genes that we saw differences in. And so this is FOXF1, which is crucial in blood vessel development. And you can see it's consistent. Ductus has a higher signal, the aorta has a lower signal, and it's consistent across the sample. And this is frizzled, which is a modulator in the Wnt pathway, which is important in cell differentiation, migration, very important in embryology. And you can see again, it's significant. So that's what we're finding. And at the basic level, we're seeing differences in the cytoskeleton and the extracellular matrix, different genes that are expressed. And clinically, I know that, right? Because I can feel the difference with my forceps. I can feel the difference between the tissue. I know inherently that structurally they're not the same. So what we found is that there's decreased expression of elastin and collagens in the ductus and the transition zone. Okay, that's not super novel. But the transition zone has high levels of certain other genes like cardiac actin, which is different than the aorta and from the ductus. And there's increased expression of injury response, profibrotic, extracellular matrix genes, and cell junction genes in this tissue. And so all of these things point to this being a unique zone that's having a very different signaling pathway. And these are all the different signaling pathways that are involved. And I'll just mention that this idea of compartment boundaries and the crossover, I'm working on one end of the aorta, but there's a lot of work on the other end of the aorta in Stanford looking at dissection and TGF-beta on that end. That is another border zone that is involved in aneurysm formation. But then these are the other things that are involved. There's increased expression in angiogenesis and transition zone and the ductus, and the frizzled is involved. So we can see all that genetic expression. Wanted to further verify. So this is a type, this is a proprietary technique, which I won't say what it is, a way of looking at RNA. Okay, and so it is in situ hybridization. So essentially, this is another segment of coarctation, and we're probing for different ones of those genes with antibodies. That's to see at the different levels of what's what. And so this first row here is the ductus. This is the first row is the aorta, second row is the ductus, and then this is the transition zone, and it's consistent. Okay, so it's not just from what's coming out of the tissue via the spatial transcriptomics. A second technique is confirming the same presence of different mRNA across the board. And then the next level, if you just continue to follow the dogma, for the most part, is you have DNA expression, you have transcript mRNA, and then you go to protein. So is the protein expression the same thing? And so this is an example from one patient. So we did what's called laser capture microdissection, and that is where you use a laser and you cut out this section, whatever it is that you are interested in. So I cut out the ductus, I cut out the transition zone and the aorta, and then did mass spec on it. You digest it, and then you do mass spec on it in order to look at the different proteins. And you can see, while less, not as much at the mRNA level, there are differences between proteins between the transition zone, the aorta, and the ductus. We are in the process of analyzing that thus far. But what you can see is this is a heat map of those peptides, and it's a lot of the same things that come up. So tenascency is obviously a big player. As you look at these are, so pink is aorta, blue is ductus, and yellow is transition zone. You can see that there are some differences, but a lot of things are similar, really showing that there are smooth muscle cells in the transition zone. It is not just a dead zone or an area of extracellular matrix, but there are what are characteristic smooth muscle cells that appear to be more or less normal, but they have a different function. Something else has been turned on in them. And then if you look at the ECM and basement membrane, you can also see that there are some very stark differences. I don't know if we can see that, but tenascency is a big player in all of this, or I think will be a big player in all of this. But the main point thus far is that there are smooth muscle cells that are doing this and driving this work. And so what have we been able to conclude from all of this? But these are the genes that are different in between the different tissues, and these are the launch points for further investigation to see can an animal model of coarctation of aorta be created, and what is the exact pathway of involvement of these different things. Most importantly, this area is a unique area. It isn't just ductal cells, which is more or less what people have thought for a long time, that's just ductus that is in the wrong spot and constricts, etc. No, it's a unique smooth muscle cell that has had the different tasks, and that will help us understand the why of that. So we have multiomic evidence that this is unique. and that this differences in transcriptome represent a breakdown in the ductus and the aorta, and it's a breakdown of the compartment boundary. So I know this is like might be a new concept. No one's ever heard of compartment boundaries. Probably, I doubt you've heard about them in medical school, but this is something that's coming, and I think this is the basis for a lot of congenital hurts. I think where these zones during embryology come together and have signaling problems where there's a breakdown is part of the pathobiology of how congenital heart surgery forms. And it's probably more important for other pathologies than we actually appreciate. And so what are we doing next? So currently we are in the process of doing laser capture and microdissection of the rest of those samples I told you we were trying to use to confirm our transcripts. So my collaborator from UConn comes out and we do this as a lab because this is a very specialized technique. And so this summer, that's what we're working on. We're gonna sequence the rest of our transcripts, of our patients currently with more traditional techniques of single cell that can give a deeper read of that signal. And then, since I've been here, which is almost 4 years coming September, we're going to look at all the other areas of coarctation that there are. So initially I was looking at just the neonates, those kids that were so significant that within like 3 days, 5 days, they had to be operated on because it was such an extreme pathology. But there's kids that present at 2 weeks. There's kids that present at 5 years old. I think the kid that I'm doing this afternoon is like 9 days old. Like there's a whole variety of ages of these kids, and it has to do with how severe their lesion is. And so I think there's some difference in that. There's a spectrum. It's not, they're not all created equal. And so we're looking at that now. And then we're going to be analyzing our hypoplastic arches because I think the coarctation signal is very important for why the rest of the arch doesn't grow. And so we'll be looking at that. And all of that is like new and me hypothesizing, not proven. And that's the fun aspect of doing this sort of thing is you're actually not making it up, but you have an idea and you get to go try and sort it out in the lab. So why the impact of all of this? So why is this important? Because it's answering the why, right? Like, we're using, and Seattle Children's and UW are very unique in being in Seattle where a lot of this technology was developed. And we have a very unique multidisciplinary team of a bunch of different experts who can all work together to solve this problem. Mark, my collaborator, had been dabbling in this area and is a very accomplished vascular biologist who more or less set the theory of compartment boundaries. But he wasn't able to, cross over and make more conclusions because he never had access to human tissue or to anyone else who could explain, that's not how it works or this sort of thing, because there was no one clinical. When I started, I had all the clinical and a little bit of the basic, but I didn't know the depths of embryology or vascular biology. Like I was not trained in that. I can't tell you in the mouse embryo, like at what day this signal happens, but Mark can. And so us working together allows us to have way more insights than you would individually. And then building the rest of the team of those data analysts that I was telling you about have all worked for the company that created this technology. That's the great thing about living in Seattle. So when that company laid off a bunch of people and stuff, I snapped up their scientists for my basic science research. So we have this very unique team and we're quite poised to advance the field. And so we're going to dominate this little region and hopefully have a biomarker in the future that'll impact how we treat this particular disease in the future. And so, like I said, minimal competition in this area because most people are tired and just want to operate, make money, and see their kids at this point. But things are set up here in such a way to promote science, which is very, very important. So it allows me to have time to do this. So, however you go through, whatever area it is that you want to go through and work in, don't give up on trying to figure out the why. Because if we don't do it, it doesn't get done. If you haven't noticed, it's been, this operation was invented in 1944. There hasn't been a lot of advancements. We still cut the two ends and sew it together. Like, that's not very novel. And so I encourage you to think about what it is you want to do. And so then this gets me back to the other point I wanted to make because of just the climate in the world right now. So the importance of being a surgeon scientist. Most of the advancements that we know as surgeons, bypass machine, operating on things, those were done by surgeons, people who were clinical in the operating room, had questions as to the reason why, and then took that back to the bench and innovated. The idea that we should just go through and operate and not think more or try more is I think a setup for disaster for us as a specialty and as a community. And I think that it's very important for us to be involved. I'm not saying that you have to like operate all day and then go to the lab at night, but being involved to collaborate with the people who are in the lab all the time is super important in order to advance the field, because you would think that there would be more understanding of what we do, but there really isn't. And people can't, and a lot of basic scientists get stuck because they don't understand basic concepts that are obvious to us as clinicians. So you're very valuable to different teams. And I would just encourage you to get involved in that aspect. And then the other reason I think it's super important to build both of these facets for those who are interested is that, so I know this is a very famous quote from Michael Jordan. So I've missed more than 9,000 shots in my career. I've lost almost 300 games. 26 times I've been trusted to take the game-winning shot and miss, and I've failed over and over and over again in my life, and that's why I succeeded. So failure in surgery is a little hard, right? So I can't exactly fail over and over as a congenital heart surgeon, because then babies die, right? You don't get that. You're not really allowed at this point to iterate. People of the past, when they were inventing procedures and stuff, they were allowed to really, suss it out and be able to fail, and the world was forgiving. That's not really the world we live in right now as a junior surgeon, as a surgeon in general. But if you go to the lab, you can try and fail. You can practice, you can work on it. So you want to be like a fantastic coronary surgeon? Work on ligating the thoracic aorta in a mouse. Like you want to be really skilled? Spend 2 years operating on rats. You want to be a great critical care doctor? Keep all the sheep alive. Like, that's not an easy task, okay? Like, there are places to be able to hone these type of skills. It's not always necessarily in the operating room, in the hospital where you're dealing with a live patient. But the idea and the practice of failing, figuring out, regrouping, trying again is incredibly important for your development as a person and as a surgeon. So I think it helps. I don't know how many of you were athletes in the room, but that whole, like, when you're in high school, the student athlete, how student athletes are like super sought after by colleges, or that idea. And why was that? It's because you can handle your academics and you do something else competitive or physical or whatever it is, but you're able to do both. And one informs the other, right? I became a better student because I was a very good athlete, and I was a better athlete because I was a student, right? They help each other. It builds you as a person and gives you skills to deal with stuff. And I think it's the same thing about being a surgeon and about being a scientist. You're constantly thinking and evolving. And so these are some of my soapbox things that I was like, oh, I'm going to talk to a bunch of residents. Let me share some things that I think are important. And then, this is one of those other concepts that good judgment comes from experience, and experience comes from bad judgment. But like I said, it's very difficult in this day and age for people to go through that process. And so the lab is a place where you can do that, okay? Because it's not with patients, it's either with animals or X, Y, or the other thing. And this also, I think, underscores the importance of those mentors. You don't find a mentor because you ask someone to be your mentor, right? That's like in kindergarten where you ask people if you want to be friends. That's not how it happens, right? It just evolves naturally. And there are people who will take you under your wing and teach you about stuff. So I didn't understand how to run a lab. Mark Majeski and Nino and the other people down at the Seattle Children's Research Institute downtown were very important to my development in that area. And they didn't have to, but they did it because someone did it for them. So Mark was trained and inspired by a vascular surgeon here, Alec Klaus, who was here for many years. And he's one of his, mentors who helped him when he was initially becoming a scientist. And so, Mark, who's like a full professor and is at the tail end of his career, to me, is doing the exact same thing. He's helping set me up, teaching me the ropes about how to write grants, how to put the papers together, how to run your lab, how to reiterate, don't give up, okay? Like, you want to talk to people who fail a lot, talk to basic scientists, because it doesn't always work. Most of the time, it doesn't work. Applying for grants where you spent, the last 7 months putting together a grant only to have it be shut down without any comments and rejected. And then you have to try again because that's how you fund yourself. Like, those are all very good lessons in how to fail and will serve you well. But this judgment is very important, and it is hard to acquire just always in the hospital. So I have a little video which I like. I don't know, I'm sure many of you have seen it before, but it kind of speaks to this point. So I'm just going to play it. Ooh. Hold on. I don't hear anything.
Exceptionally thorough. Your hat had to be perfectly starched, your uniform immaculately pressed, your belt buckle shiny and void of any smudges. But it seemed that no matter how much effort you put into starching your hat or pressing your uniform or polishing your belt buckle, it just wasn't good enough.
Now, maybe I should just preface this. I'm a child of a colonel in the Army. I married a naval aviator. There's a lot of military in my family. So this sort of stuff speaks to that. I was in the Army for a short stint before I decided I couldn't kill it. So there we go.
Several times a week, the instructors would line up the class and do a uniform inspection. Several times a week, the instructors would line up the class and do a uniform inspection. It was exceptionally thorough. Your hat had to be perfectly starched, your uniform immaculately pressed, your belt buckle shiny and void of any smudges. But it seemed that no matter how much effort you put into starching your hat or pressing your uniform or polishing your belt buckle, it just wasn't good enough. The instructors would find something wrong. For failing the uniform inspection, the student had to run fully clothed into the surf zone, then wet from head to toe, roll around on the beach until every part of your body was covered with sand. The effect was known as sugar cookie. You stayed in the uniform the rest of the day, cold, wet, and sandy. There were many a student who just couldn't accept the fact that all their efforts were in vain, that no matter how hard they tried to get the uniform right, it went unappreciated. Those students didn't make it through training. Those students didn't understand the purpose of the drill. You were never going to succeed. You were never going to have a perfect uniform. The instructors weren't going to allow it. Sometimes, no matter how well you prepare or how well you perform, you still end up as a sugar cookie. It's just the way life is sometimes. If you want to change the world, get over being a sugar cookie and keep moving forward.
So I think.
several times a week.
So I think that's one of the things I wanted to convey is you don't learn to fail. It doesn't come naturally to anyone, right? None of us, are happy when we don't get what we achieve. But your endurance as a surgeon is going to be determined by how well you can deal with your failures and how you regroup. And I think one of the best places to learn how to do that as you become a resident, and one of the spaces where you're allowed to do that is in the lab. And I think it will help you immensely as you go forward as a surgeon and change your perspective and how you think, and will help our field grow and advance in whatever area that you're interested in. But I would just throw down that gauntlet and say that get involved, try, don't give up, and just forget that it's our job to advance the field, to figure out the why, and to answer the questions. This is my family. You don't have to give up everything to do that, okay? You just have to manage your life just like you would as a student athlete or a surgeon scientist or anything else. But those are the things I wanted to talk to you guys about today. Thank you very much for your time, and I'm happy to take any questions.
Dr. Greene, this is such a great July grand rounds. I want to say a couple of things that I noticed and a question. So thank you for reminding us about congenital disease and congenital heart disease, because those of us that aren't at Seattle Children's aren't thinking about it all the time. So I think it's great for the faculty, for the residents, for the students to kind of remember what they were seeing in medical school and think a little bit about congenital heart disease. But your points about being a surgeon scientist is inspiring, because it's so hard. It's hard to do that now. And even before January 20th, it was hard to do it. And it's even harder to do it now. So thank you for showing us the ways that it can be done, and the aspects of collaboration and the people that you develop, and the willingness to fail to ultimately succeed. So I think it's inspirational. Really appreciate you and the presentation. The question I have relates to the work that you're doing, and I'm going to ask about it in two directions. So before birth, because everything you're doing is essentially relates to at birth, what's happening. So before birth, do you see that this might be leading to interventions that we could do in utero that could impact congenital heart disease or other congenital disorders, that we could impact them before delivery and before we have to do an operation on them? And the second half of that is, the vast majority of us are adult surgeons. We have adult vascular surgeons here. How do you see that this also might then affect decisions or management of disease in adult patients?
Thanks, Dr. Wood. So to answer the first question, yes, I do think that is where technology will lead us, but we're not there, right? We are obviously like several steps away from being at that point, but that's the goal. Once we are able to figure out the pathway, identify the genes or proteins that are involved in this, then therapeutics can come into play. And the goal would be to prevent these sorts of problems from happening, because it is something, I believe it is something early in embryology. I don't think it is just random. I don't think someone, patients and families will think all sorts of things. Is it because my neighbor smokes a lot of pot, and because of that, like I got, this or that, or did this, glass of wine matter, or X, Y, or Z thing, as they wonder why their child has this. But I think as we understand more, then that would be the goal, would be to prevent disease, just like in other aspects. And with regard to the adult world, I think that this technology is not limited to the congenital space by any stretch of the imagination. Like the advancements that have been happening in this area of the world are massive in the last 10 years. And it is going to allow for so much more rapid identification of different processes. So I would say if you're in the adult space, really think of what question is it that you are trying to answer first. And then talk to different people, because these technologies are much cheaper, much faster, much more accessible than they used to be back in the day. Like when I was in college and, trying to do a DNA microarray was a huge to do. And now, your turnaround is so much faster and it's so much easier than it was, 20 years ago.
I have a comment from the Zoom. This is from Dr. Mogul. Excellent talk, Dr. Greene. I know you scratched only the surface of the body of work you have successfully and unsuccessfully worked on, but speaking to us about this really underscores the importance of why the importance of the why and of what we do. And so thank you for that. And if there's, I have a question if nobody else. So you mentioned a few times that there haven't been many advancements or changes in the actual surgical procedure over the number of decades. Has any of your research or the work you've done and that balance between being a scientist and the surgeon, not that I plan to do any kind of patent or anything, but do you have any ideas of how, or any kind of adjustments or advancements in the technique, understanding that maybe if all this goes forward, maybe we won't really need the technique much anymore?
From our end, really, our contribution has been making it very clear that you need to do a coarctectomy during a Norwood. We put that data out there because there are a lot of, not a lot, but a handful of contingents who still don't really understand that, accept that, and so they have a higher recurrence rate. Because in order to do a coarctectomy, you have to cut the aorta, two ends of the aorta, and then you have to sew the two ends together. So that takes more time. It's more circ arrest time. It's more clamp time. And so people don't do it because of ease and things like that. But what we found is that long term, it's actually detrimental to the outcome. So I think that's one aspect. Not necessarily in my basic science work, but in my engineering work, yes. So Seattle Children's is one of the world leaders in blood conservation and bloodless surgery. And so my engineering work revolves around trying to figure out the optimization of the bypass machine. And so there are a couple intellectual property things on that aspect of my work.
One of the concepts that comes up, other states, the aortic syndrome, that's the concept. That work on the heart. Look anything on the aortic syndrome.
Yeah, your interest area. Yeah, no, obviously I stay above the diaphragm for the most part, but yes, that's very ripe with because as we've mentioned, the compartment boundaries are throughout. That's how we formed as humans. And so no, I don't do anything like that, but if people have ideas, I know a whole host of people who would be willing to collaborate and help if you thought you had a region that was important.
Just one last comment in this last minute. This is from Dr. Calhoun. As mom to two sons with congenital heart disease, keep fighting the fight. While it can be frustrating, heartbreaking, and forgive the pun, hard, it is ultimately fulfilling. Thank you for all you do. And thank you for your presentation.